Okay. Hi everybody, my name is Nick Anderson. I'm a fourth year PhD student in the cancer biology theme of the Graduate Biomedical Sciences program here at UAB. And today I'm going to tell you a little bit about what I do in my research. So I study acute myeloid leukemia, which is a type of blood cancer. Cancer in general is when you have some group of cells that are growing way more than they should. So the human body is a fascinating thing. All your cells in your whole body are very well controlled in how much they're supposed to grow or when they're supposed to stop growing. And what happens in cancer is some of those controls get messed up. So the cells grow faster than they're supposed to, and that's how you get a tumor, because there's too many cells growing and growing and growing when they're supposed to stop. Now, leukemia is a little bit different because it's not a solid group of cells in a tumor, it's your blood cells. They're growing way more than they should. So you just have lots of extra cells circulating in your blood and in your bone marrow. So all your blood cells are made in the bone marrow. You have lots of different kinds of blood cells. You have blood cells that fight infections. You have red blood cells that carry oxygen so that your whole body can work. You have platelets that clot, so when you get a cut, you stop bleeding. And all of those come from the bone marrow, from what are called hematopoietic stem cells, or blood-forming stem cells. So what happens in cancer of the blood is you get these stem cells, or some of the other cells that help make all the blood cells. They grow more than they're supposed to, and they stop differentiating or turning into those functioning blood cells that actually do good things. So one of the ways that we look at leukemia is with what's called a blood smear. So I have here some blood samples that I've taken from one mouse that has leukemia and one that doesn't. And I'm going to show you how we prepare these smears so we can look at them under the microscope and see if they have leukemia or not. So we take a microscope slide, and then we take this micropipette here. And we want to mix this up so all the different cells are all mixed together so we don't get a whole bunch of one kind and not a different kind. And we take just a little tiny bit, put it on this slide. And the reason it's called a blood smear is because we actually smear the blood. So I'm going to take a second microscope slide. And I'm going to use this to streak the blood across that slide. So now we got it kind of spread. I don't know if it's easier to see with or without that, but so it's spread across that slide now. And then what we do is we'll let that dry for about a half hour. And I've got these other ones that I smeared about a half an hour ago. So the first thing that we do after they've dried is we put them in methanol, which fixes the tissue, or basically like chemically freezes it so the cells stop changing, stop moving, and then we can go on to stain them so we can look at them under a microscope. So these ones I've already fixed in the methanol. And then we put them in this special stain called a Right Gemsa stain. This is specifically made for doing this type of staining for blood cells. So we put it in this one for about a minute and a half. And that's got a kind of bluish purple stain and a red stain that stain different parts of cells that I'll explain when we get onto the microscope here. But another thing that we use these smears for is to tell if our chemotherapy is working. So I'll show you in a second what a leukemia blood smear looks like and what a non-leukemic blood smear looks like. So we use that for diagnosis, but then after we've treated a patient or a mouse, we look at the blood smears again and we look and see if we've gotten rid of the leukemia cells on the smear. So this is really, really useful tool that I use a lot in my research to see if what I'm doing is working, if I'm curing leukemia. So something that I'm really interested in specifically for my project is when a lot of patients, when we give them chemotherapy, it works really well at first, but then they relapse or the disease comes back really quickly. And so there's a lot of research going on as to why that happens. What's going on that makes some of the leukemia cells not die and come back? So one thing that we think is important is the other cells in the bone marrow. So your bone marrow has lots of different kinds of cells, not just the ones that make blood. You have cells that make bone, you have cells that make fat, you have cells that make cartilage, you have nerves, you have blood vessels. So there's lots of other stuff going on in the bone marrow. So my hypothesis for my project is I think that there's a specific type of cells in the bone marrow that's protecting these leukemia cells. So when we treat the mouse or the patient with chemotherapy, it kills any other cells that aren't protected in this little, what we call a niche, this little pocket that they're hiding in. So what I'm trying to do is figure out what type of cells that is, how we can make those stop protecting the leukemia cells so that when we give the patient or the mouse chemotherapy, we can kill all the leukemia cells so they don't relapse, so the disease doesn't come back. Alright, so now that's been staining for about 2 minutes, but that's fine. So then we put it in water to kind of wash the stain off. And then once that sits in the water for a minute, take it out and let it dry. And then it'll look like one of these. So these, hold it against the white lab coat here, you can probably see it better. So these are all purple now. So these ones are from a group of mice that I treated with chemotherapy or a placebo to try to see if this treatment was working. So this particular mouse here had leukemia, but then we gave it chemotherapy. Now, what are we looking at here? So these little funky looking circles are red blood cells. So almost all the cells here are red blood cells. And then these big fat purple ones are actually white blood cells. So these ones are the ones that fight infections, the T cells, the B cells, the neutrophils. The red blood cells are the ones that carry oxygen. And then we've also got, if you can, I don't know if you can see it on the video, but there's these little tiny purple dots. Those are the platelets. Those are the ones that help clot. So when you have leukemia, you get a whole bunch of these, but not enough of the other ones. So you bleed, you don't have enough oxygen, and these ones are actually, they don't work to fight infections either. So you get infections, you don't carry enough oxygen, and you don't clot. So really, really bad stuff. But this mouse that was treated with chemotherapy, the chemotherapy actually worked really, really well. So you see not very many of those big fat purple cells. So now let's look at a mouse that has leukemia. So this one was given a placebo, or basically just like sugar water without the actual chemotherapy drug in it, so that we could have something to compare to. So this one looks a lot different. See, there's lots of these big fat purple cells and almost no platelets and fewer red blood cells. Now, you can probably tell a lot of these big fat purple cells, they look different from each other. It's because there's a lot of white blood cell types. So when a patient goes in to see if they have leukemia, they get a smear like this. They take it to a hematopathologist. That's somebody a doctor who's been trained to know what blood cells look like under a microscope. They'll do what's called a blast count. So they take some sort of counter like this, and it may be hard to see with the lights down, but there's a little counter here for each type of blood cells. So there's a counter for basophils, eosinophils, and there's going to be a test at the end. You have to know exactly what all of these do. Just kidding. So then they'll look at a smear, and they'll say, okay, this one is a neutrophil. Count one of those. There's 3 more neutrophils. This one's a lymphocyte, and they'll count 100 or 200 total cells, and then count up all these numbers and calculate percentages. Now, this is like an old crusty thing, so now there's an app for that. That's prettier and easier. So when I do these smears, I just use this app that I can use to count if they're leukemia cells or normal healthy cells of different types. So now we're gonna increase the magnification a little bit so we can really see the differences between some of these cells. So what I'm doing now is I'm adding an immersion oil. So, little physics lesson, when light is passing from one medium to another, or from air to glass back to air, it bends a little bit. Now that refraction, or that bending in this microscope, when it goes from air to glass, back to air, back to glass again, that bend is actually larger than the resolution that we want. So when we put this oil on there, the oil actually touches the glass of the lens, so it doesn't have to bend as many times. The light bends from air to glass, and then there's very little bend between the glass, the oil, and then the glass on the lens. So we can actually get a much higher resolution, excuse me, much higher resolution image. So if you come back over here to the computer screen, we can see much more clearly what these cells look like. So here's a good example of several different kinds of cells here. So the big purple blob in each of these cells is the nucleus, and then you can see the cytoplasm is the lighter purple. Now we've got here, this is not a very healthy looking one, but it's a neutrophil. So that is the type of cell that normally fights infections. This one here is what we call a monocyte. So this one actually eventually will make one of these, but you shouldn't see very many of these normally. So this is actually a sign of leukemia. And then these ones over here are much larger. The nuclei are weirdly shaped. These are all leukemia cells, or blasts. So when a doctor is diagnosing leukemia or checking to see if chemotherapy works, they'll go through this slide and they'll count. They'll count up all the leukemia cells. So leukemia cell, leukemia cell, leukemia cell, leukemia cell, healthy neutrophil, healthy neutrophil, healthy neutrophil. And then they'll get a percentage at the end, and they'll say this patient has 20% blasts, or 80% blasts, or 5% blasts. So what we're looking for when we treat is a lower blast count. So if the patient had 95% blasts and then they get chemotherapy and they have 4% blasts, then we know that chemotherapy worked. So now what I want to do is put another slide on here, and then anybody watching can tell me if they think it's leukemia or not leukemia. All right, you ready, hematopathologists? So let's go back over to the screen here. All right, let's scroll through here. So what do these look like? Obviously, these are the red blood cells. These look like maybe neutrophils. These also look like maybe neutrophils, but that one looks like a leukemia cell. But overall, there are not very many white blood cells, so this looks like a mouse that got chemotherapy that worked pretty well. So let's put up another one, see what we think. So this one also looks pretty good. Not very many white blood cells. Those look like they could be leukemia cells, but again, not very many. So this one looks like it got chemotherapy, and the chemotherapy worked pretty well. You see, these are the normal, healthy neutrophils or infection-fighting cells. So now you're ready to go diagnose leukemia. So my ultimate goal is to figure out, like I said, a way to make our chemotherapies work better for more patients. So hopefully in a couple of years when I'm finished and I'm writing my thesis, I will have more to tell you about that. But yeah, anybody watching have any questions?
So, is there a difference between the cells depending on if it's like AML or ALL, like different types of cancer?
Yes, good question. So AML is acute myeloid leukemia. ALL is acute lymphoblastic leukemia. So when I was showing here all the different kinds of blood cells, you can have a leukemia that originates from or comes from any one of those different cell types. So an ALL is one that comes from lymphocytes. So when you look at them on a smear, these leukemia cells will look more like lymphocytes, but they'll be dysplastic, which basically means they're not fully formed. They're not quite done making a mature, functional, working lymphocyte. When it's acute myeloid leukemia, those leukemia cells will look more like these myeloid cells, the neutrophils, the monocytes, the myelocytes. So there is a morphological difference, or they look different in their shape, the size and arrangement of their nucleus. And I'm not a certified expert in that. Like I said, doctors who go to medical school and train for years are the ones who can really, really tell those differences on the blood smears. But yeah, that's actually how they can tell if it's AML, ALL, CLL, CML, by looking at the shape and how the cells look on the microscope.
So what's the time frame in terms of when you look at one of those smears and it's like covered in white cells, and then they get chemotherapy and then it clears up? Like what's that? How long does that take?
That's a good question. So it depends on the type of leukemia. So there's different types of treatments for different types of leukemia, but for AML, it's generally a couple of months. They'll go through a couple rounds of chemotherapy treatments before they get a really good response like that.
How did you get into this kind of research?
So I actually originally became interested in cancer in general because I had a grandfather who passed away of cancer, and it just really interested me how cancer works. And my goal is, if someday some grandfather who has cancer, some grandmother who has cancer gets treated and lives long enough to meet their grandkids that they wouldn't have otherwise met, then I will have succeeded. But so I kind of had a general interest in cancer, but didn't really know exactly how I wanted to pursue it until actually I was in a high school biology class. Mr. Jones, honors biology, he took us on a field trip to the Fred Hutchinson Cancer Research Center in Seattle, and we got to go through some of their research labs and hear somebody talk about their research. And we extracted DNA from strawberries, which are hexaploid. They have 6 copies of every chromosome. We only have 2. It's kind of interesting. But yeah, being in a research setting like that just fascinated me. So that kind of motivated me to go more towards the research route rather than like to be an oncologist, be a doctor treating patients. And then just the more classes I took during college, the more interested I got in cell biology, how cells are different, and the molecules that they use to interact with each other, and how the disease develops at a really, really subcellular level. It's just really fascinating to me.
Is there a difference in cancer cells in children and adults?
Yes. There are a lot of ways that those cells can be different. On a smear, they'll look really similar, but the way that cancer comes about in adults and children is often very different. But that's a lot harder to tell from just looking at a microscope.
Now, so if you were looking at like a human blood sample, would it look different than from what you were looking at right here with the mice?
Yes, a little bit different. Pretty similar. So let me put one of these back up here. So red blood cells in mice and humans look almost exactly the same. They're almost exactly the same size. They're about 8 microns wide. So a meter is about 3 feet. 1/1000th of a meter is a millimeter. 1/1000th of a millimeter is a micron. So really, really small, 8 microns. In mice, the neutrophils, those healthy, good, normal infection-fighting cells, the nucleus is a little funkier looking than in humans. In humans, it'll usually have fewer lobes or like lumps to it. And then in mice, it's normal for them to have mostly neutrophils and fewer lymphocytes, but in humans it's more common to have more lymphocytes and fewer neutrophils in a healthy, normal individual.
So the general concept is still the same.
Yeah, very similar. Is another question coming? Okay. Well, if there's any other questions, I can go on to Facebook later and respond to them with typed out answers, but yeah, that's what I do in a nutshell. Hopefully in a couple of years I will be able to say I did it, I cured cancer. I don't think that's going to happen, but that's the goal. So thanks for watching. Have a good one.